In this study, we aim to understand the role of ApoA1 mimetic peptides as potential therapeutic agents for promoting cholesterol efflux in the cell. So a little bit of background about ApoA1. So ApoA1 is a peptide which wraps around HDL particles and helps stabilize it, as well as helps it act in the body. So HDL has been widely known as the quote-unquote good cholesterol, and when delivered in the body, it can go around and scavenge for cholesterol and actually efflux them from different cells. So what we did is we studied the last sequence, the last helix of ApoA1, and we actually, as you can see here in table 1, is we did different kinds of methylations to it. So we had A alpha, D alpha, K alpha, and L alpha, and 6 alpha that we created, which compared to the control had more methylations all across of it. Not only that, as we also calculated the hydrophobic moment with different kinds of hydrophobic moment calculators, and we were able to see that except for the A alpha and D alpha, the K alpha, L alpha, and 6 alpha all had increased hydrophobic moments. And this translated to what we saw is in our lipid solubilization assay, which is a good measure for cholesterol efflux capacity. Our A alpha was unable to clear the DMPC vesicles versus our 6 alpha and D alpha and alpha and K alpha were all very good at clearing the vesicles. When we actually did our cholesterol efflux assay, what we saw was the best cholesterol effluxers peptides were the 6 alpha, K alpha, and L alpha, as you can see in figure 3A. Not only that, these 3 peptides were also able to form the pre-beta HDL, which is the more nascent form of HDL and leads downstream to the more mature form of HDL, which again, remember, has cardioprotective effects in the body. Not only this, these peptides, when compared to the control in figure 2, you're able to see that they're resistant to digestion by proteases such as trypsin and APS-M. This is something that needs to be further studied, and also proves very important because current peptides are unable to achieve this as well. So overall, what we did is we methylated our peptides, and this led to proteolysis resistance, as well as the ability to efflux cholesterol effectively from the cell, as well as form new pre-beta HDL. Hopefully this will lead to cardioprotective effects as we further develop these potential therapeutic agents. Thank you.